Hello and welcome to Meiosis for Dummies. If you're here, you're having trouble in your high school biology class because you don't know what meiosis is or how the heck it works. Well, I've been there before and luckily I'm here to teach you. Meiosis is the process that somatic cells go through to create gametes. When you're there, you're already lost. Gametes are sex cells or cells responsible for reproduction. Boys, you have sperm cells in your testes. And girls, you have eggs in your ovaries. No, not that kind of egg. An oocyte. The sperm cells are a lot smaller than the oocytes because they need to swim at really fast speeds to reach the oocyte.
Just keep swimming, just keep swimming, just keep swimming, swimming, swimming.
The oocytes are very big because they don't need to move much except for dropping when a woman ovulates. To remember that plants have sex cells too. That's right, plants have sex. And anyways, before we can get started on the beautiful process of meiosis, we need to go over a few important terms. A haploid is a cell that only has one set of chromosomes, such as the sperm cell or the egg cell. A diploid is a cell with two sets of chromosomes, such as most body cells or a unfertilized zygote. Homologous chromosomes share the same gene loci in the same position. Sister chromatids are two identical copies of a replicated chromosome. Well, that is all you need to know, so
let's get down to business.
Meiosis happens twice. That is, it happens in two phases. One phase is to split the homologous chromosomes, and the other is to split the sister chromatid. The result is 4 daughter cells as opposed to 2 that are created by mitosis. While all 4 sperm cells are usable, 3 out of the 4 oocytes do apoptosis, meaning they die for a purpose. And the purpose they die for is to ensure the survival of the 4th oocyte. Before meiosis can begin, there is something that needs to be done known as interphase. All chromosomes are duplicated in a process similar to chromosome replication before mitosis. Inside the nucleus of animal cells are 2 centrosomes, each containing a pair of centrioles. The 2 centrosomes are produced by the duplication of a single centrosome during premeiotic interphase. Once the chromosomes are replicated, it's officially time for meiosis to begin.
It's the start of something new.
It all starts with something called prophase. During the first prophase, the duplicated homologous chromosomes pair and crossing over occurs. Crossing over is the process that can give rise to genetic recombination. At this point, each homologous chromosome pair is visible as a tetrad, a tight grouping of 2 chromosomes, each consisting of 2 sister chromatids. The sites of crossing over are seen as 2 crisscross non-sister chromatid and are called chiasmata. Next up is the first metaphase. The pairs of homologous chromosomes, now tightly coiled, line up in the center of the cell, equidistant from both poles. Spindle fibers from one pole of the cell attach to chromosomes for each pair, and spindle fibers from the opposite pole attach to the homologous chromosome. Next up comes anaphase. Anaphase begins when 2 chromosomes of the tetrad move towards the opposite poles. A key difference between mitosis and meiosis is that the sister chromatids remain joined after metaphase in meiosis 1. Whereas in mitosis they separate. Now it's time for telophase, the last step in meiosis 1. Homologous chromosome pairs complete their migrations to opposite poles of the cell. Now a haploid set of chromosomes is at each pole, with each chromosome still having 2 chromatids. Nice! A nuclear envelope reforms around each chromosome set. The spindle disappears and cytokinesis follows. In animal cells, cytokinesis involves the formation of a cleavage furrow, resulting in the pinching of the cell into 2 cells. After cytokinesis, each of the 2 progeny cells has a nucleus with a haploid set of replicated chromosomes. That's it for meiosis 1, but we all know meiosis happens twice. So let's start this all over again. We're going back to prophase. While chromosome duplication took place before meiosis 1, no new replication is necessary before meiosis 2. The centrioles duplicate. This occurs by separation of the two members of the pair, and then the formation of a daughter centriole perpendicular to each original centriole. The two pairs of centrioles separate into two centrosomes. The nuclear envelope breaks down. Guess what? What? Then metaphase happens again. Each of the daughter cells complete the process of forming a spindle apparatus. Then, just like in mitosis, they line up along the metaphase plate, in contrast to metaphase 1, in which homologous pairs of chromosomes align on the metaphase plate. If you were paying attention last time, you know it's now time for anaphase. The centromeres separate, and the two chromatids of each chromosome move to opposite poles of the cell. The separated chromatids are now called chromosomes in their own right. Oh my god, we're almost done. It's time for the final telophase. Cytokinesis takes place, creating 4 daughter cells with haploid sets of chromosomes. Because of crossing over, some chromosomes are seen to have recombined segments of the original parental chromosomes. And that's it for meiosis. But there are still a few things we need to discuss before I can let you go. Because the whole purpose of meiosis is to create genetic variability, that is, to ensure that every single person has a different set of genes, a few things need to happen to guarantee this. I briefly touched on this earlier, but during prophase of meiosis 1, the double chromatid homologous pairs of chromosomes cross over with each other and often exchange chromologous segments. This recombination creates genetic diversity by allowing genes from each parent to intermix, resulting in chromosomes with a different genetic complement. Another way genetic variability is increased is due to something called random assortment. Crossing over in meiosis 1 leads to non-identical chromatids in meiosis 2 chromosomes. During anaphase of meiosis 2, the centromere joining each chromatid pair dissolves, creating 2 chromosomes of each type. The outcome of which chromosome will go to which gamete is random, so that each gamete has a potentially unique combination of genetic material. But sometimes, in meiosis, not everything goes as planned, and something called a disorder occurs. The most common disorder is called non-disjunction. Non-disjunction occurs when the chromosomes fail to separate properly as the cell divides. The result of non-disjunctions are gametes containing the wrong number of chromosomes. In humans, this would lead to a person containing 45 or 47 chromosomes. 60% of miscarriages are due to this tragic thing called non-disjunction. Well, I'd hate to leave you after talking about how mothers lose their babies because sometimes meiosis is dumb, so enjoy this for a minute.
It's a piece of cake to bake a pretty cake. If the way is hazy, you gotta do the cooking by the book. You know you can't be lazy.
Okay, I'm feeling much better, and I know you must be too. Well, I hope you learned a lot, and this has been Meiosis for Dummies.